This AI just discovered a way to make you live longer. A new study was just published by scientists that claim they have found a way to slow down aging with artificial intelligence. And it's got people talking because this is one of the first studies to show how an AI can discover new medical knowledge independently. So what did they find? What does it mean for aging research? And what should you do about it? I'm a board-certified dermatologist and health educator. Let's talk about this. All right, so the title of the paper is pretty straightforward. It says exactly what it means. Deep learning identifies molecular signatures of aging in single cells from the human body. So this is a group out of the Buck Institute for Research on Aging. They had this AI look at various scientific data sets and then try to find patterns and connections that humans might not have seen. So here's the deal. The reason we don't have a cure for aging or a cure for age-related diseases like cancer and dementia is because we don't fully understand aging itself. Like we know our cells have a lifespan. We know that when cells divide, their telomeres get shorter and shorter until eventually the cell can't divide anymore and then it dies. But there are other reasons why cells die before they reach that Hayflick limit, which is the number of times a cell can divide. Cells can be damaged by toxins or carcinogens, meaning something that causes cancer, or they can have trouble fixing their DNA after being exposed to UV light or other sources of radiation. Also, cells can run into problems during cell division when the DNA gets tangled or damaged. All these things can lead to cellular senescence, meaning the cell stops dividing and enters this state of permanent growth arrest. And we think that these zombie cells contribute to aging. So if we want to figure out interventions for slowing aging, we need to first understand what makes a cell senescent in the first place. To do that, researchers analyzed blood samples from 10 individuals across different age groups. The participants were 3 healthy young adults, 3 middle-aged adults, and 4 older adults. The blood samples were taken during routine visits where the participants donated blood for other research projects. In this study, they only used the leftover blood samples and de-identified the data so no one could figure out who the samples came from. Now this is important because this is an example of using data from others, which is an important part of open science. When researchers put their data sets online for others to use, they can speed up progress in aging research without having to repeat experiments that have already been done. So, back to the study. They took the leftover blood samples and isolated the immune cells called peripheral blood mononuclear cells. Then they used a technique called single-cell RNA sequencing to see which genes were turned on or expressed inside each individual cell. Essentially, all cells in the human body are identical, except for the fact that some cells do different jobs based on which genes they turn on. So, for example, your skin cells have different genes turned on than your brain cells. And that's what makes them look and function differently. The RNA sequencing was able to identify 5 distinct cell clusters. These are 5 different types of immune cells. Then they looked at the gene expression patterns within those clusters to identify differences between the youngest and oldest groups. They wanted to figure out which cells were more senescent in the older individuals. And they found that 2 of the immune cell clusters had significantly more cells from the older donors compared to the younger donors. This suggests that these 2 immune cell types play a role in aging. And then they wanted to find out why these cells were senescent. They used something called gene set enrichment analysis to compare the older group to the younger group. This analysis identified certain pathways and biological processes that were more active in the older group. And what they found was that the most significant difference was an increase in the immune response to viruses. This suggests that these 2 cell clusters in the older group were trying to fight off viral infections. And this is interesting because normally, when you get a viral infection, your body will clear the virus and then return to normal. But these immune cells in the older donors didn't seem to be able to clear the viral infection. Instead, they stayed activated even after the virus was cleared. Researchers also noticed that these same two cell clusters had lower levels of certain genes, including a gene called DUSP22, which encodes a protein that regulates inflammation. When they knocked out DUSP22 in young mice, they developed more age-related health problems, including inflammation, hair loss, osteoporosis, and a thinner skin. So it seems like DUSP22 may protect against aging. All right, so now let's talk about the AI. As I mentioned earlier, researchers used a type of artificial intelligence called deep learning to analyze the gene expression data. Deep learning is a branch of machine learning that uses multiple layers of processing to extract features from data. So in this case, they used a type of deep learning algorithm called a deep neural network. The neural network was trained on a large dataset of gene expression data from various tissues and cell types. After training, the neural network was able to accurately classify cells as senescent or non-senescent based on their gene expression patterns. So here's how it works. First, the raw data was pre-processed, meaning they cleaned the data and made sure it was in a format the AI could use. Then the data was split into training and testing sets. The training set was used to train the AI, and the testing set was used to evaluate its performance. The AI was then trained to classify cells as senescent or non-senescent based on their gene expression patterns. After training, the AI was tested on the testing set to see how well it could classify cells. Okay, now let's talk about the results. The AI was able to accurately classify cells as senescent or non-senescent with over 80% accuracy. It also identified the top genes that contributed to the classification, which were mostly the same as the ones that the researchers had identified through their own analysis. This suggests that the AI was able to capture the key features of aging cells and that it's comparable to traditional methods for identifying senescent cells. In addition to identifying the top genes associated with aging, the AI also created a heat map that showed how the expression of these genes changed over time. This heat map identified two clusters of genes that were highly correlated with aging. One cluster included genes that were upregulated or turned on during aging, while the other cluster included genes that were downregulated or turned off during aging. This suggests that the AI can not only identify the key features of aging cells, but it can also uncover new insights into the molecular mechanisms of aging. Okay, so what does this all mean? This is an exciting development in the field of aging research for several reasons. First, it provides new insights into the molecular mechanisms of aging. By identifying the genes and pathways that are associated with aging, researchers can gain a better understanding of the biological processes that drive aging. Second, it offers a potential new way to identify and target aging cells. By developing drugs that target these aging cells, researchers may be able to slow down the aging process or prevent age-related diseases. Third, it showcases the potential of AI to accelerate scientific discovery. In this study, AI was able to identify key features of aging cells and uncover new insights into the molecular mechanisms of aging. This suggests that AI has the potential to revolutionize aging research and help us develop new interventions for slowing aging and age-related diseases. So this is just one more step in the long journey to figuring out how to slow down aging. This was a small study. There were only 10 participants and they were all Caucasian. So more studies need to be done to confirm these findings in larger and more diverse groups of people. However, this is still an important first step towards understanding the molecular mechanisms of aging and developing new interventions for slowing aging and age-related diseases. If you want to learn more about aging, check out my playlist on healthy longevity. If you enjoyed this video, please leave me a thumbs up and subscribe to my channel. And if you want to support my work, consider becoming a patron. Thanks for watching and take care.